chapter 17, Metabolic Test Part 2. So we're doing follow-up on some of our tests. And you all will remember we first did our IMViC tests. Okay. So help me out, you guys. What does the I stand for?
Indole.
Indole, good. And what's the M stand for?
Methyl red.
Methyl red, good. The V?
Something that I can't pronounce.
I know. Voges-Proskauer and the C?
Or something.
Citrate. Good. Okay. So we want to explain why this series of tests were developed. So we have to go back to our whole great concern with fecal contamination of drinking water and food. And you guys remember, if we have fecal contamination of food or drinking water, possibly a great variety of enteric pathogens could be present. We could have, for example, toxin-producing strains of E. coli. We could have Salmonella, Shigella, Vibrio cholera. We could have polioviruses. We could have hepatitis A viruses. We could have noroviruses. We could have many of the intestinal protozoal parasites we've discussed. Giardia, Balantidium could be present. Entomobacillolica could be present. We could have worm eggs present. We could have ascaris and tinea eggs. So this is why we're so concerned with fecal contamination of food and drinking water. Now, many of those pathogens are either hard to grow or hard to see. So we want an organism that's easy to grow in the lab, easy to identify in the lab, to be our fecal indicator, right? And you guys remember the bacterium we said was going to act as our fecal indicator?
E. coli.
Who that was?
E. coli.
E. coli, right. Okay, so we use the presence of large numbers of E. coli in food or drinking water is an indicator that possible there's fecal contamination and thus any of those other pathogens could be present. And we're like, oh no. Now the problem is E. coli has many cousins, and one of them, a close cousin, is called Enterobacter orogenes. Enterobacter orogenes belongs to the same family as E. coli, the Enterobacteriaceae. It's a gram-negative bacterium, it's motile, it's oxidase negative, it ferments lactose with gas production. It looks really similar to E. coli. But the natural history of Enterobacter orogenes is different. It too may be found in the intestinal tract, but it also can grow well in the environment. It can grow in dead, decaying organic material like plant material. So if we find large numbers of Enterobacter orogenes in drinking water or in food, and we don't see many E. coli, it might mean that it's just natural runoff from surrounding soil. And it doesn't necessarily mean that we got fecal contamination. Okay, so the question is, if these two guys are so closely related, they share so many similar properties, how do we quickly distinguish them apart? And the answer is the IMViC test is one way we can quickly distinguish E. coli from Enterobacter orogenes. And the reason is, these two cousins give the exact opposite results in the IMViC tests. Okay, so this is something you're going to want to memorize, you guys. So just before you go to sleep at night, when you get up in the morning, just memorize this. So the IMViC reactions for E. coli are indole positive, methyl red positive, Voges-Proskauer is negative, citrate negative. Okay, so if you know E. coli results and you know Enterobacter orogenes is the opposite, what can you tell me about Enterobacter indole?
Negative. Negative. Positive. Positive.
Dynamite, you guys. So what we hope you guys are going to see today, that your E. coli and your Enterobacter orogenes are going to give these exact opposite IMViC results. Okay. Now you already ran the indole dry slide. So did most of you get a positive indole in E. coli?
Yes.
And negative on Enterobacter? Yes. Good. Okay. So what we need to do then is we're going to follow up on these last three results. Now, instead of writing a lot of the directions on the board, I'm going to refer you to the page number in your lab manual. And the reason is, after we get back from break and we take our lab practical number 2, we're going to start our unknowns. You're going to get 2 unknown bacteria. And you're going to use everything you've learned to identify your bacteria, including these metabolic tests. Now, the unknown experiment, it's open book, open notes, open computer, but you can't ask your colleagues for help. You can't ask a two-legged mammal for help, okay? And one of the things that I've noticed, you know, and this happens every semester, is a lot of times folks are reluctant to actually use or read the lab manual. You know, they'll you know, it's good. They listen to what we say. They look at what we write down. It's good. They ask their neighbors if they have any questions. But for the unknown experiment, you guys, you have to be able to use your lab manual. And so just to stress that, what I'm going to do is put the page numbers of how to run the tests up on the board. And I'm going to ask you guys, let's read the lab manual. Let's find the information together to make sure you know how you're going to use the lab manual when you do your unknowns. Okay? So let's do the methyl red test. So in my version of the lab manual, it's described on page 144. Okay, so you guys remember that you inoculated your high glucose methyl red medium, and let me see here. Let me just go grab some samples back here really quick. We have set up some practice guys for the metabolic test. Okay, so you all remember that you inoculated your methyl red media, which is high glucose media. What is the methyl red trying to detect? High acid reduction from which fermentation pathway? Mixed acid fermentation, right? So we have our little cartoon over here, you guys, showing two different fermentation pathways. Mixed acid fermentation, where we get a mixture of acids, hence mixed acid. We get a really low pH below 4.4. So we inoculate our broth, we incubate our organism, and so I'm going to pull my tubes out of the incubator, and I'm going to look at my tube and go, doesn't look like anything happened. What did I just do wrong?
Did add the indicator.
Yeah, remember, and that happens every semester. Folks forget that after incubation, you have to add what to the tubes before you read them? Methyl red, right? Okay, remember the methyl red would kill the bacteria if you added it ahead of time. So we have to let them grow, and then we add the methyl red indicator. So the methyl red pH indicator is up front. Be careful, you guys. We're going to have so many different little brown bottles running around, and often folks will put the wrong bottle in the wrong container. So even though, you might think, oh, this is where the methyl red is, when you pick up the container, what do you always do? Read the label, okay? So I just verified I've got my methyl red. So now what do I do, you guys? I've got my methyl red, I've got my tube. Now what do I do? Where do I look to find out what I should do next? In a lab manual. You guys are like, come on. But it's true, you guys. I want you to know where you can find this information. Okay, so help me out, you guys. How do I run the methyl red test? Help me out. Can you guys help me? What do I do? And I want you to help me by looking in your lab manual, okay? What do I do?
144.
Seriously, you guys, it helps so much if you know where to find this information. Because remember, when we do the unknowns, nobody can help you. Okay, so look at the directions for running the methyl red test. I've got my incubated tube. I've got my methyl red. What do I do now? Yes, yes, good, good. Okay, so you should have your goggles on for this. Ooh, I'm being a bad instructor. Whenever you're working with these reagents, remember your mom would tell you the reagents will burn your eyeballs out. So you want to put your goggles on, and to each of my tubes I'm going to add, how many drops? 4. Okay, 4 drops. All right, so we're going to add 1, 2, 3, 4. Okay, if mixed acid fermentation occurred and the final pH is below pH 4.4, what color should my broth be? Red. Remember, methyl red turns red with low pH, pH 4.4. Okay, so help me out, you guys. Is this a positive? Is this a positive or a negative test? It's negative. It's actually negative. Okay. So, what does a negative test tell us?
Lacks enzymes.
It lacks the enzymes to carry out mixed acid fermentation. Dynamite, you guys. So this would be a negative test. What about this one? That's positive. That's a positive test, okay? So what does this tell you about your organism? Right, it has the enzymes to carry out mixed acid fermentation. Dynamite, you guys. Excellent, okay? And of our E. coli and Enterobacter orogenies, this would be which one?
E. coli.
E. coli, right. And this would be?
Enterobacter.
Dynamite, you guys. Excellent. Okay. When you're finished reading your tests and nobody in your team needs them anymore, please take all the tape off, barely loosen the caps, and we're going to put them in the kill area, okay, in the broth portion. Dynamite, you guys. Okay. The next one is the Voges-Proskauer test, and it too, the description is on page 144. Okay. Now this is the nasty one, you guys. This is the nasty one. So remember, if you're pregnant, you're trying to get pregnant, you think you might be pregnant, or if for any reason you don't want to work with these chemicals, all be your very kind of slow robot, right? So I'll be your hands, but you have to tell me exactly what to do. So let me kind of walk through this with you guys. So first of all, we need to be in full safety gear. So I have a wild variety of gloves. Okay, so you want to make sure the person that's actually doing the experiment, you want to make sure that they're going to glove up. They're going to have their safety goggles on, their lab coats on, and we want to have our lab shoes on. Okay, so what I'm going to do is pull my tubes, and let me see here. I'm going to grab one. Okay, so what we'll do is we'll put a sign-up sheet. Wow! Oh dear. We're going to put a sign-up sheet on the board for the safety hood, because we want you guys to run this in the safety hood. So you'll take your tubes from the incubator, you're going to carry them over here to the safety hood, and you'll place them in the test tube rack. Oh, excuse me. Okay, so I'm going to bring my tubes over here. We'll get another test tube rack for you guys. Okay, so I'm going to put my tubes here in the safety hood, and then I want to make sure that I have my reagent. So you guys know we need two reagents, VP reagent A and VP reagent B. If you guys ever work in a lab, never let people tell you, oh, use reagent A, reagent B. You want to know exactly what those reagents are, right? That's important to protect yourself. So what is the identity of Voges-Proskauer's reagent A? Alpha naphthol. This is nasty stuff, you guys. So this is the one we want to be want to be careful with those. Let me bring this back over here. We buy the alpha naphthol commercially prepared. And the alpha naphthol is in a little glass vial inside this plastic dropper bottle. And the plastic dropper bottle already has a hole in the tip. So what we're going to do is, I have really weak fingers, you guys, so I have to do this two-handed. I'm going to take and hold the alpha naphthol, the VPA reagent, in between my thumb and index finger here. And I'm going to, for me, because I'm so weak, I have to do a sandwich with my other hand. And what I'm going to do is I'm going to point the tip away from myself. I don't want to go like this. I don't want to go like that, right? And what we actually want to do is point it towards the corner of the safety hood away from ourselves. And we want to do one squeeze, a single squeeze, and you'll hear the glass tube crunch. Don't continue to grind it, because you'll make little tiny glass particles that will plug the dropper tip. So one crunch, and then you want to tap it, and that will bring the glass particles away from the dropper tip. And then I'm going to add my alpha naphthol to my first tube. And help me out, you guys. In the directions, how many drops do I add? 15 drops, right? And this, you guys, is so hard. These little things are so hard to squeeze. But just be patient. Okay, so you just add it drop by drop.
It says 15.
2, 3, 4, 5, 6, etc. Okay, so we'll pretend that I've added my 15 drops. Now what do I do, you guys? Do I read it now?
Mix gently. Mix gently.
Okay, am I going to put the cap back on and hold it and gently mix it, okay? Make sure the cap is on securely. You don't want it spilling all over yourself. Now what do we do? Now we're going to add 5 drops of VP reagent B. What is VP reagent B? 40% potassium hydroxide, right? And it will burn your eyeballs out, just like the mom.
Burn
So Val and Carmen have prepared the 40% potassium hydroxide. As always, you guys always read the label in case somebody's put the wrong bottle up here. So 40% potassium hydroxide. How many drops do I add?
5.
5, okay. So I'm going to add 5 drops. Now what do I do?
Let it sit for 15 minutes.
I'm going to go ahead, tap it tightly to mix it. Okay. And now I want to loosen the cap, right? We want to have air. We want air to have access. I'm going to leave my tubes up here for up to an hour. And I'll check my tubes about every 15 minutes. Okay. If your organism carried out butylene glycol fermentation, they will make a unique intermediate called what? Acetylene, right? If acetylene is present, the alpha-naphthol 40% potassium hydroxide react with it, and within an hour you can see a beautiful
Red.
a beautiful red color. If your organism is Voges-Proskauer positive, what does that tell you about your organism? Kind of like, so what? Who cares? What does it tell you about your organism? If your organism is EP positive.
It's usually methyl red.
Yeah, it uses butylene glycol fermentation pathway. I know this sounds kind of silly. You guys are like, well, of course, but these are how we word it on the lab practical. So I just want you to get used to what answer. Okay. Now, so let's say it's been an hour, you guys. Here's another tube. Is this EP positive or negative? Negative. Okay, good. So what does that tell you about your organism? It doesn't carry out butylene glycol fermentation. Good. All right. So of your E. coli Enterobacter-Rogenes, who would this be? You would predict. E. coli. Good. And who would this be? Enterobacter-Rogenes. Dynamite. Okay. Okay. Now here's the thing. Now unfortunately the previous lab section didn't do this, but we really want to help the folks that are helping us. So please, before you leave today, make sure you take the tape off your tubes. We're going to leave them in the hood. The VP gets through this chemical hazardous waste. But if we don't take the tapes off, it really takes hours for the folks that are helping us to take it off. So we want to help them, right? So don't do as the previous lab sections did and left their tape on. That's not cool. We want to help the folks helping us. So please just make sure you take your tape off before you leave today, okay? But leave them in the hood. Hazardous waste procedures. Also, folks, the containers of alpha-naphthol, try to use them up if you can. Like, for example, with this one, maybe I have a little residual. What I'll do is I'll look at the board and see the next person. I'll call their name. When they come up, you just ask them. It's like, I have a little bit of alpha-naphthol remaining. Would you like to use it? Ideally, we want to use it up if we can. When you're finished, we have these very low-tech waste cans, and it just says EPA waste. So you just put your little dropper bottle in there, because that too has to be treated as chemical hazardous waste. And as always, you guys, we want to protect each other's backs. If we see our colleague is up here without gloves, without their goggles on, just remind them gently, we're trying to help one another. And just as a little reminder, we mentioned during the unknowns that you guys can't ask one another for help, but there is an exception. If you see your colleague is, doing something dangerous, or you see they've forgotten an important safety precaution, like you see them running the BP test without their goggles, their gloves, don't hesitate to tell them. Or if you feel uncomfortable telling them, just come tell me. Whenever safety is involved, we talk to one another, right? We try to help one another. So during the unknowns, never hesitate to tell someone if you're worried for them, if you think they might be doing something that could hurt them or hurt others. Or like I say, if you're uncomfortable, just come and ask me, okay? And remember, we're just trying to help one another. So that's the exception to the rule with regard to the not asking others for help during the unknown. Okay, you guys, we got one more on our IMViC test. Which test is that? Citrate test, right? Okay, which media did you inoculate to run the citrate test? Citrate agar, right? And remember, it's that beautiful medium. Why is it green? What's the pH indicator? Bromothiamol blue, right? Okay. Now remember, you guys, you streak the slant, you grew these guys aerobically. If your organism can use citrate as a sole source of carbon and energy, if it has the enzyme citrase, what will happen to the pH of your media? The pH goes up, right? What color is bromothiamol blue to alkaline pH?
Blue.
Oh, that's so beautiful, you guys. This is like my favorite test. Isn't that gorgeous? That's sometimes described as Prussian blue, or some folks say Levi jeans blue, but that's a positive citrate test. Okay,
Wow.
so we get an alkaline reaction that bromothiamol blue turns. Bromothiamol blue turns blue. Okay, so that's lovely. Would we predict this would be E. coli or Enterobacter?
Enterobacter.
Enterobacter, that's right. And so if the organism can't use citrate as a sole source of carbon and energy, your medium would be
Green.
Okay, so who would this be, E. coli or Enterobacter?
E. coli.
Good, you guys. Excellent. Good job. Okay, so we're hoping you're going to get these same results, all right, for your test. Now, again, with your Simmons citrate agar, when you're finished with it, please take all the tape off, barely loosen the caps. I'm going to put them in the kill area, right? Okay, just one more to go, you guys. Is it hot in here, or is it just that time of my life?
It's cold.
All right, you guys, the last thing we're going to do, and this is so cool, is the TSI agar slants. So let's go through all the questions for the exam. What does TSI stand for? Triple sugar iron. Good. What are the triple sugars? What are the three sugars? Glucose, lactose, sucrose. The same ones we use in the PR sugar fermentation tubes. Dynamite, you guys. What does the I stand for?
Iron.
Iron, right? So we have iron salts in the media. What's the purpose of the iron salts? Now remember, you guys, it's so good to guess. Guessing is really a good way to learn. Good. I'm hearing it, you guys.
Break down the sulfur.
Okay, it will combine, the iron salts will combine with which toxic molecule that your microbes might make?
Sulfide.
Hydrogen sulfide,
Yeah.
right? Remember from lecture we said hydrogen sulfide, it's a cytochrome inhibitor. It'll shut down your electron transport chain, which might not be good. So we don't want you sniffing your tubes to see if your organisms made hydrogen sulfide, you know, that rotten egg smell. And we don't want to kill you. You know, we need you guys out there to save us old folks later on. All right. So we put the iron salts in there to combine with the hydrogen sulfide to make what? Iron sulfide, right? And iron sulfide, how does it appear? Black. Okay, and oh my goodness, you guys. Your Proteus read the lab manual. I'm so proud. Look what your darling Proteuses
Oh,
did.
wow.
Isn't that amazing? That is a huge amount of iron sulfide. That tells you your Proteus was pumping out the hydrogen sulfide like crazy. Okay, another possible question, you guys, on the lab practical. Can you describe 2 ways your microbes can make hydrogen sulfide? Why did we stab the butts? What was one reason we stabbed the butts? To get them to grow anaerobically, right? So, do you know of a way that microbes can anaerobically produce hydrogen sulfide? Anaerobic respiration, right? Using what is a terminal electron acceptor? Sulfates, right? Okay? So that is a great way that some microbes make hydrogen sulfate. They use anaerobic respiration. They use sulfates as a terminal electron acceptor. The sulfates can get reduced all the way to hydrogen sulfide. Okay, so that was one reason we wanted them growing anaerobically in the butt. That was one reason we wanted you to stab the butt. Let me sweep this line. Good. A second way they can make hydrogen sulfide is how?
Aerobically.
Yeah, they can use. Some of them can use sulfhydryl-containing amino acids as an alternate source of carbon and energy. So when they strip off that sulfhydryl group, it gets released as what? Hydrogen sulfide, right? So that's the second way they can make hydrogen sulfide is through the breakdown of sulfur-containing amino acids, like cysteine is a great thing. Good job, you guys. Excellent. Okay. So to answer the question, can your organism make hydrogen sulfide, we're going to look for that black precipitate. And this is incredible, you guys. Be aware, sometimes all they'll make is a little bit of black in the butt. They're so good. I just want to give them a hug. They're just but sometimes it's just a little bit in the butt. This is really dramatic, okay? So the iron sulfide tells us they made what?
Hydrogen sulfide.
Hydrogen sulfide, right? The iron sulfide tells us they made hydrogen sulfide. That's just a little bit quirky. We just got to remember that, okay? All right. So this is our TSI cartoon up here. There's a lot of different results. So you want to remember the black precipitate is iron sulfide. It tells us the organisms made hydrogen sulfide. Now, in addition, remember we said there's 3 sugars in there, glucose, sucrose, lactose, and there's a pH indicator present. What's the pH indicator, you guys, in TSI? Ooh, I heard it. Phenol red. Good.
Yeah.
Phenol red's a pH indicator. And the phenol red will let us determine if those sugars were fermented, right? So if the sugars are fermented, they make acids, the pH drop. What color does the phenol red turn? Yellow. Good. Okay. So let's look at the colors of phenol red and see there's 3 different possibilities. Okay. If your organism cannot ferment any of the sugars, just as we saw in the PR sugar fermentation, they'll use the proteins, the amino acids, as an alternate source of energy. And when organisms are breaking down amino acids, the amino group gets released as what? Ammonia, right? And we know that's a weak base. So what happens to the pH? pH goes up, right? What color is phenol red at alkaline pH? Red, okay? So if your organism can't ferment any of the sugars, the butt will be red. And the slant will be red, okay? Now, okay, and this is a little bit tricky, and I don't want to mess with you more than need be. Okay, so I'll just describe the appearance, and then those of you that want to know the why behind it, we can get together later, and I'll try to describe the why behind this. Okay, so deep breath, guys. All right. If your organism can only ferment glucose, you'll have an acid butt, not you, but the tubes will have an acid butt, which will be yellow, and you'll have an alkaline slant, which will be red. Okay, so if your tubes have a yellow butt and a red slant, that tells you only glucose was fermented. Okay, only glucose was fermented. In contrast, if your organism can ferment glucose and at least one of the other sugars, either lactose or sucrose, or possibly both, you're going to have an acid butt and an acid slant. So the butt will be yellow, the slant will be yellow. Another thing you're checking for is the presence of cracks or bubbles in the agar. Why might you have cracks or bubbles in the agar? Gas production, and usually we're looking for molecular hydrogen or carbon dioxide. Okay, so let's take a look at your darlings here and see if we can't figure out what the heck's going on. Okay, so let's take a look at your Proteus. Oh my goodness, what a great bacterium that is. Now this is a challenge, you guys, because if we ask you about sugar fermentation, there's so much iron sulfide in the butt you can't see is it red or yellow. So here's something you must memorize. When you get this much iron sulfide produced so you can't read the butt, you presume the butt is acidic. You presume it's yellow. And it's something you just have to memorize, right? So with this Proteus, we can't read yellow or red of the butt. We presume it's a yellow butt, and therefore we presume glucose was fermented. Okay, glucose was fermented. Now, we can read the slant, and the slant is what? What color is it? It's red, right? That's alkaline. So we've got this reaction going on. So which sugar or sugars did Proteus ferment? Glucose only. Good job, you guys. All right. Now let's take a look at your E. coli. And some of you will get different results, okay, but here are a couple of E. coli results. It's like, did somebody do anything wrong there? Did they inoculate these incorrectly?
No.
That's all the gas the E. coli were making from fermentation, okay? So you see that's a lot of gas. So let's go ahead and read these guys. So was hydrogen sulfide produced? No, there's no black iron sulfide. So E. coli does not make hydrogen sulfide. That's one observation. Now let's read sugar fermentation. Is the butt acidic?
Yeah.
Yes. So we know which sugar was fermented?
Glucose.
Glucose, right? Is the slant acidic?
Yes.
Yes. Yes. So we've got this reaction right here, right? So we know that glucose was fermented, and we know at least one of the other sugars was fermented, either lactose or sucrose, or possibly both. From your PR sugar fermentation test, you guys know which sugar was fermented. Of lactose and sucrose, which sugar did E. coli ferment in your PR fermentation tests? Was it sucrose or was it lactose? Remember, E. coli likes to live in the intestinal tract of mammals who drink milk, and milk has
lactose.
lactose, right? Yeah, okay. So E. coli fermented lactose in our test, but if you remember, it didn't ferment sucrose. Okay. Now, just looking at this TSI slant, we wouldn't know that, right? All we know is they fermented either lactose or sucrose or both. We would have to run the PR sugar fermentation test to know which one of the sugars they fermented. Okay. And again, you guys, why this space here? Why is the agar blown apart like that?
Gas.
Gas production from fermentation. Now, be aware, you guys, sometimes it won't be this dramatic. Sometimes it might only be a crack or a bubble in the agar itself. But again, these guys really were studying their lab manual. I mean, these are just so dramatic, you know. But this is how we like it for the lab practical. We want really dramatic results for you, okay? Okay, so I think that's it. So with these, you guys, when you're finished, please make sure to take all the tape off, loosen the cap slightly, put them in the kill area. We've set up some practice, like kind of like pretend lab practical stations in the back, just to give you a little bit of practice on these guys.